हेलो फ्रेंड्स, एज वी नो दैट योर एंट्रेंस एग्जाम इज नियर बाय। हम भी तो आपके लिए कंसर्न हैं, इसीलिए इस वीडियो में हम लेकर आए हैं 50 मोस्ट एक्सपेक्टेड क्वेश्चन which has came into the last 5 years in the history of DU entrances. Let's begin. A halter is a vestibular organ of butterfly, modified forewing of beetle, balancing organ of a housefly, or organ used by a male insect to attract female for mating. See, halter is a club-shaped organ of flying insects, which provide information about body rotation during the flight, like just like a gyroscope, and hence it is a balancing organ of a typical diptera. So option C would be the correct answer for this question. Which of the following are schizocoelomates? Platyhelminthes, Annelida and Mollusca, Aschelminthes, Annelida and Orthopoda, Annelida, Orthopoda and Mollusca, Orthopoda, Mollusca and Echinodermata. First of all, you should know what is schizocoel. It is a body cavity which is present between the digestive tract and musculature of body wall, and it is formed due to the splitting of mesodermal mass. Remember, there are three group of people which are the classical example of schizocoelomate, which we can remember as a mnemonics arm AAM: Annelida, Orthopoda, and Mollusca. So option C is the correct answer in this case. Exon skipping is associated with nonsense mutation, promoter mutations, splice site mutations, or silent mutation. Before we answer, let's understand what is exon skipping. Basically, if you see in the given figure, we have taken an example of dystrophin gene. This dystrophin protein, it is being made up of because of the expression of various exons. So if all the exons are being processed and translate a protein, then the functional protein will be formed. Somehow, if suppose some of the exon is missing, like in the other case of your right hand side, if you can see the exon 2 is missing. So the protein will be formed, but it is a truncated version of the fully functional protein. Now, is it a defective protein? Yes, it is truncated, but still it is a functional protein. So even though there is a exon is missing, but yet it is not causing a much problem in a truncated protein. Hence, the exon skipping is associated with splice site mutations in this case. So option C would be the correct answer. In bivalves, which structures secrete pearl? Whenever any foreign mass comes and is stuck up between shell and mantle of bivalves, they start secreting layers of nacre, which is also known as mother of pearl, and which ultimately start forming the pearl. Hence, in this case, the option C is the correct answer. Pearl mother layer in mollusk is, as we have just discussed in the previous slide, it has to be the nacre. That means option B. Match list with list 2 items. List 1 contains the organism while list 2 contains the larva. See, you need to remember a lot of larva of different organisms, because this is a pet question which comes in the exams where you have to match the following, or they are going to ask you a different larva of different trophic levels or even organism. In this case, if we see, the hexaconth larva belongs to Taenia, the Glochidium larva is belonging to Unio, Planula is belonging to Obelia, Tornaria larva is for acorn worm or Balanoglossus, while Miracidium larva is of Fasciola. Hence, in this case, the option C would be the correct answer, that Taenia is having hexaconth larva, Unio is having Glochidium larva, Obelia will be having Planula larva, while Balanoglossus is having Tornaria larva. To make it simple, we are also giving you a table where you can learn a different phyla and their classical larval form. True and complete metamorphosis is observed in First of all, we should know what is true or complete metamorphosis. If an insect is having all different looking or phenotypically or morphologically looking different forms during their life cycle, then it is called as complete metamorphosis, and such insects are called as holometabolous insects. Three major order for this type of insects are dipteran, lepidopteran, and calyptrons. Calyptrons means beetles, lepidopterans means butterflies and moths, and dipterans means mosquitoes and your houseflies. So in this case, the option A is has to be the correct answer. Which of the following, again the complete metamorphosis हमें बताना है, as we have just discussed. Lepidopteran, that means butterfly and moth. Calyptron, that means beetle. So option C is the correct answer. And then dipterans like flies and the mosquitoes, they shows the complete metamorphosis. So option C here would be the correct answer. Insects such as Drosophila undergoes three molds before undergoing metamorphosis. Molting is controlled by which of the following hormone? That's very easy. Ecdysone is also known as molting hormone, which governs the molting processes. So option B is the correct answer. The term tunicate make reference to the urochordate test or tunic, which is composed of. See, tunicates or urochordates are the only animals known to produce and composed of crystalline cellulose. So in this case, the option A is the correct answer. In terrestrial vertebrates, which of the following structure did not arise from the pharyngeal pouches? Eustachian tube, middle ears, invertebral disc, or parathyroid glands. Basically, in tetrapods, when they develop, so pharyngeal clefts, they produce parts of ears and other structure of head and neck region. For example, first pharyngeal pouch, it led to the development of eustachian tube or middle ear. Second, it led to the development of palatine tonsil, while third and fourth, they actually helps in developing parathyroid gland. Intervertebral discs, they are not made by the modification of pharyngeal pouches. So hence option C would be the correct answer in this case. The origin of jaw in the gnathostomes is from the gill arches, bone supporting the cranium, notochord, or HK pouches. Basically, gnathocoms develops jaws to capture the larger prey. So they have a very large buccal cavity so that they can capture the larger prey. And with these buccal cavities, they are developed from the simple transformation of their ancestral gill arches, especially the rostral gill arches or mandibular arches. So hence, the origin of the jaws in the gnathostomes is from the gill arches. Option A is correct. Y-shaped chevron bone is present in thoracic vertebrae of mammals, cervical vertebrae of birds, caudal vertebrae of a reptile, or lumbar vertebrae of amphibian. A typical Y-shaped chevron bone is present in the caudal vertebrae of a reptile. So here the option C would be the correct answer. How many chambers are there in the heart of a frog tadpole? 1, 2, 3, or 4? An adult frog always have a three-chambered heart, but its tadpole, which is more adapted to the aquatic life, that has a two-chambered heart with having one atrium and one ventricle. So option B would be the correct answer. Match list 1 with the list 2 items. Here in the list 1, there are different taxonomic hierarchies are given and we have to match with their list 2 components. So let's take the example here. In this case of toad, the domain is eukarya, kingdom is animalia, division is gnathostomata. That means A is connected to 3. Phylum is chordata, while subphylum is vertebrata. Class is amphibia. That means B is being connected with the 4th. Order is Anura. That means C is connected to A. Family is Renidae. That means D is connected to the 2. Hence, the option A would be the correct answer in this case. Precursor for steroid hormone synthesis is tryptophan, cholesterol, stearic acid, or glycogen. Cholesterol, it is the precursor for all steroidal hormones like glucocorticoid, mineralocorticoid, or sex hormone, which is being synthesized in different tissue. So hence option B would be the correct answer.
फ्रक्टोज इंटॉलरेंस इज ड्यू टू द फ्रक्टोज इंटॉलरेंस जो होता है वो एल्डोलेज बी एंजाइम की वजह से होता है, और जिनमें भी ये डेफिशिएंसी होती है या रिड्यूस प्रोडक्शन होता है, दे डेवलप hypoglycemia if they consume fructose or sucrose which also has a fructose, because they cannot tolerate fructose. तो इसका आंसर होगा सी, एल्डोलेज.
The major pathway producing NADPH is TCA cycle, fatty acid synthesis pathway, glycogen degradation pathway, or hexose monophosphate pathway. Basically, the oxidative phase of pentose phosphate pathway, or also known as hexose monophosphate pathway, is required to generate the 2-NADPH. Hence, option D would be the correct answer. During synthesis of ATP in glycolysis, phosphate is provided by AMP, inorganic phosphate, PRPP, or high energy phosphorylated compounds. The phosphorylation of ADP to ATP, it is carried out by the compounds who is having high phosphoryl group transfer potential, like 1,3-biphosphoglycerate or phosphoenol pyruvate. So hence the option D would be the correct answer. At isoelectric pH, a protein carries. That's very simple. At isoelectric point or a pi, the protein contains no net charge. That means it is in the zwitterionic state. So it becomes an electrically neutral. So option D would be the correct answer in this case. If cardiolipin biosynthesis decreases in eukaryotic cell, ATP synthesis would be unaffected. The function of ETC would be affected adversely. The potential would remain at 140 millivolt, or ADP would be imported into the matrix. See, as we know that cardiolipin is the chief constituting molecule for the inner mitochondrial membrane. As our electron transport chain is heavily present on the inner mitochondrial membrane, so any compromise with cardiolipin will adversely affect the electron transport chain. So option B is the correct answer. Which one of the following is wrong match for enzyme classification? Enzyme commission 1, that is oxidoreductase, enzyme commission 2, that is transferases, EC3 hydrolases, or EC4 ligases. Basically, if you can see the table, then you will recall that EC4 is basically lyses, which break any bond without the using water, and that's where EC4 has to be lyase rather than ligases, which is EC6. So hence option D would be the correct answer, that EC4 is a wrong match for the enzyme classification. Bidirectional movement of vesicles requires kinesin 1, microtubules and microfilaments, a flexible neck region on the motor proteins, or association of plus and minus end directed motors. Basically what happens is, as we know that microtubules are polarized, that means they have plus and minus ends, and that is where the specifically molecular motors like dynein, which is a negatively directed motor, and kinesin, which is a positively directed motor, they both collectively works in tandem to derive the bidirectional vesicle transform, which is also known as tug of war. So as you can see in the diagram, there are two molecular forces are there. One is dynein. This molecular motor actually is taking the cargo towards the negative side, negative end of the microtubule, while kinesin, it is taking the cargo towards the positive end of the microtubule, and hence the option D is correct, that the bidirectional movement of vesicles, it requires association of plus and minus and directed motors. Cilia and flagella, they contain a contractile protein which is called as dynein, tubulin, actin, or myosin. Basically, eukaryotic cilia and flagella, they are composed of a contractile motor protein that is called dynein, and a non-contractile microtubulin which is composed of tubulin proteins. So hence option A would be the correct answer, that the dynein is the contractile protein which actually uses the chemical energy of ATP and generate a force which is required for the locomotion or any kind of a. Gap junctions are not essential for transfer of secondary messengers, metabolic coupling, peristalsis, or skeletal muscle contractions. Gap junction, they are basically found in almost all tissues except fully developed skeletal muscle cells or mobile cells like sperms or RBC. So here the gap junction will not be essential for skeletal muscle contraction or option D. In Robertsonian translocation, fusions occurs at telomeres, centromeres, long terminal repeats or ends of the long arms. A Robertsonian translocation, it occurs when long arm of a two acrocentric chromosome they fuse at the centromere and hence two short arms are lost. So option B would be the correct answer. Which of the following technique was used by Michelson and Stahel to separate DNA labeled phase N15 from N14 labeled DNA? Isopycnic equilibrium density gradient centrifugation, agarose gel electrophoresis, molecular sieve chromatography, or ion exchange chromatography. In their classical experiment, Michelson and Stahel, they actually use two types of media, N14 and N15 rich media, to culture the E. coli. And when they try to separate the heavy and lighter form of a DNA, they use isopycnic density gradient centrifugation method. So hence, the option A would be the correct answer in this case. DNA fingerprinting technique employs unique and housekeeping genes as a probe, specific metabolic genes as a probe, VNTRs, or intron region as probes. DNA fingerprinting techniques involves identifying the differences in the number of some specific short nucleotide repeats like VNTR, which may be different in different persons. Hence, the DNA fingerprinting techniques, it employs the VNTR sequences as a probe. Southern blot analysis is used for the detection of a DNA sequence, RNA sequence, protein-protein interaction, or DNA-protein interaction. Southern blot is basically used to detect specific DNA sequences by using a specific DNA based probes in a DNA sample. So hence the option A would be the correct answer. The absorbance of UV light at 280 nanometer by a protein is largely due to the presence of amino acids with aliphatic R group, acidic R group, aromatic R group, or basic R group. As we know that optical absorption of protein is measured at 280 nanometer, which is due to the presence of aromatic amino acids like tryptophan and tyrosine. Another amino acid, that is phenylalanine, that absorbs at 255 nanometer. So at 280 we are actually measuring the tryptophan and tyrosine, that is option C. At what stage of a eukaryotic cell the chromatin will be the least compact? See, it is very easy. The chromatin will be least compact during the DNA replication, which occurs in the S phase. So hence option C, that would be a correct answer, that during the S phase the chromatin will be the least compact in nature. Cell to cell communication is important in development of an organism. The ability of cell to respond to a specific inductive signal is called, that's very easy, it is called as competence. So option B is the correct answer.
अगला क्वेश्चन है, फॉर अ गिवन फिक्स लेंथ ऑफ डबल स्ट्रैंडेड डीएनए मेल्टिंग टेम्परेचर इज, जैसा कि हम सब जानते हैं मेल्टिंग टेम्परेचर वो टेम्परेचर होता है जहां पे 50% डीएनए पॉपुलेशन इज बीइंग डीनेचर्ड और ओपन, यानी कि जो हाइड्रोजन बॉन्डिंग है वो टूट जाती है 50% पॉपुलेशन की। जैसा कि हम जानते हैं जीसी ट्रिपल बॉन्ड से कनेक्टेड होते हैं हाइड्रोजन बॉन्डिंग, जबकि एटी डबल बॉन्ड से कनेक्टेड है। सो लॉजिकली GC will contribute more to the melting temperature than in comparison to AT. तो टेम्परेचर मेल्टिंग वाला जो है डीएनए का वो बहुत ज़्यादा GC कंटेंट डिसाइड करता है, एंड दैट्स वेयर इट इज डायरेक्टली प्रोपोर्शनल टू द GC बेस कंटेंट. आंसर A has to be correct.
Bile is produced in our body which helps in controlling blood pressure, act as a surfactant to emulsify lipids in intestine, helps in digestion of starch in intestine, or helps in absorption of water soluble vitamins. Basically, bile salts, they helps in the emulsification of fat droplets in intestine. So hence, the option B is the correct answer. Import of glucose by the liver cell is dependent on hydrolysis of ATP, requires expression of GLUT1 on the plasma membrane, occurs throughout the phospholipid bilayer, or facilitated by the GLUT2. Always remember, the various types of glucose transporters are there and they are having different types of significance in different tissues. So always make a list where all the glucose transporters and their significance you have to memorize. As far as this question is concerned, the import of glucose by the liver cell is facilitated by the GLUT2. It is the most abundant and main regulator of hepatic glucose transport. So hence option D would be the correct answer. In ruminants, true stomach that is homologous to other mammals is represented by abomesum, reticulum, rumen or omesum. Always remember there are 4 chambers in any ruminant stomach. The last chamber, that is abomesum, it is an equivalent of a true stomach of any typical mammalian stomach. So hence the option A would be the correct answer.
दोस्तों, third chamber in the stomach of a ruminant mammal. जनरली स्टमक एक रूमिनेंट मैमल का जो होता है वो 4 चैम्बर्ड होता है, और अगर हम इसे ध्यान से देखें तो फूड इसमें एंट्री करता है ह्यूमन में पहले, फिर इट गोज टू द रेटिकुलम फॉलोड बाय ओमेसम. ये जो तीनों चैम्बर्स हैं, ह्यूमन, रेटिकुलम एंड ओमेसम, दे आर फर्मेंटेशन चैम्बर्स, और यहाँ से ये फाइनली एबोमेसम में जाता है जो कि एक ग्लैंडुलर स्टमक है, एक टिपिकल कार्निवोर का जो स्टमक होता है उसकी तरह, जहाँ पे कि केमिकल डाइजेशन होता है फूड का, एंड फ्रॉम हियर आफ्टर डाइजेस्टेड फूड इज बीइंग ट्रांसफर्ड टू द स्मॉल इंटेस्टाइन. तो अगर आप सीक्वेंस देखें, र्यूमन, रेटिकुलम, ओमेसम, ओमेसम शुड बी द करेक्ट आंसर. ओमेसम इज द थर्ड चैम्बर ऑफ द स्टमक ऑफ अ रूमिनेंट मैमल. यस इट इज. आगे चलते हैं दोस्तों.
Hemophilia is an example of a trait that is carried as an autosomal dominant, autosomal recessive, sex-linked dominant, or sex-linked recessive. Hemophilia is a sex-linked recessive disorder which may be inherited by a carrier mother to her son. Male cannot pass this defect to his progeny. Hence, the option D would be the correct answer, that hemophilia is an example of a sex-linked recessive disorder where the defective gene is present on the X chromosome. Hemophilia is due to a defective gene which does not produce fibrinogen, hemoglobin, thromboplastin, or prothrombin. Hemophilia is a type of blood disorder in which, due to a defective gene, a person is not able to produce enough of clotting factor, which is definitely required for stop bleeding. Hemophilia A, B, and C, which are a common form of hemophilia, they are happening due to the absence of factor 8, factor 9, or factor 11 respectively. Deficiency of factor 2, which is also known as prothrombin. It also leads to another form of hemophilia in which a person is not able to form the factor 2 clotting factor, and hence bleeding doesn't stop. This kind of deficiency is often seen in those communities where the marriages between close relations are very common. So hence, in this case, the option D would be the correct answer. Which system is active under stress? Parasympathetic nervous system, sympathetic nervous system, central nervous system, or somatic nervous system? Basically what happens, parasympathetic nervous system and sympathetic nervous system, they both constitute the autonomic nervous system, which is majorly involved in dealing with the various body process. Sympathetic nervous system, it generates flight or fight response whenever body is under stress. So hence option B would be the correct answer. Wallace line is present in between Oriental and Australian region, Ethiopian and Oriental regions, Neotropical and Nearctic regions, Palearctic and Ethiopian regions. Wallace line is a faunal boundary drawn by British naturalist Russell Wallace in 1859. It separates basically the biogeographical realms of Asia and Australia. That means Oriental and Australian region. Hence, option A would be the correct answer. Which of the following gases was probably absent at the time of origin of life? Hydrogen, oxygen, methane, or carbon dioxide? This is very simple. Since at the time of origin of life, there was a no photosynthetic pigments were there, which was responsible for delivering the free oxygen. Hence, since was an absence of photosynthetic organism, hence oxygen would have not present at the time of origin of life. So option B would be the correct answer in this case. Kidney of the vertebrate embryo develops from. This is very simple. The kidney of a vertebrate essentially develops from the intermediate mesoderm, which lies between the somite and the lateral plate mesoderm. So hence the option C would be the correct answer. That intermediate mesoderm is responsible for generating the kidney as well as gonad. While the chorda mesoderm, it allows the development of a notochord. Paraxial mesoderm, it allows the development of a head and somites, while lateral mesoderm, it allows the development of a splanchnic or somatic or extra embryonic tissues. Hence, the option C would be the correct answer. Sympatrons are co-transporters that transport small molecules and gases in the same direction, cations and anions in the opposite direction, sodium ions and glucose against the concentration gradient, and glucose against its concentration gradient. First of all, you should know that a sympator will be a co-transporter if at all it is transporting two chemicals or two metabolites in the same direction. Sim means same and co means there must be at least two components which will be transported. For example, a sodium glucose transporter which transport the sodium as per its concentration gradient, means from higher concentration to the lower concentration gradient. Definitely there will be a force will be generated which will drive the movement of glucose as per their uphill concentration gradient, that means from low concentration to the higher concentration. And that's where in this case the option D would be the correct answer, that a sodium glucose co-transporter or sympator, it derives the glucose against its concentration gradient. Import of glucose by the intestinal epithelial cell is dependent on hydrolysis of ATP, require expression of GLUT1 on the plasma membrane, occurs throughout the phospholipid bilayer, or facilitated by GLUT2. We have just discussed in the previous question that the import of glucose by the intestinal epithelial cell is heavily dependent on the hydrolysis of ATP, which ensure the working of sodium potassium ATPase. So option A is the correct answer. Which of the following is observed during anaphase A? Depolymerization of microtubules, shortening of polar microtubules, consumption of ATP by motor proteins, or sliding of polar microtubules. See, one thing is very clear, that whether it's a anaphase A or anaphase B, during anaphase event, the chromosomes, they get separated and tries to go towards the poles. Here the activity of these spindle fiber tubulins, it is of very much importance. There are three types of tubules are there. The kinetochore microtubules are those to which the chromosomes are attached. Polar microtubules are those spindle fibers where there's no chromosomes are attached, and there is another type of microtubule is there which is present at the pole, that is called astral microtubule. So during the anaphase, whether it's a one or two, activity of these microtubules that causes the separation of chromosomes and movement of these chromosomes towards the respective poles. So exactly what happens during the anaphase A? There is a depolymerization of kinetochore microtubules happens, and because of which there is a shortening of this microtubules happens, and this derives a force by which the chromosomes are forced to get separated and move towards the pole. This very same kind of a thing happens during the anaphase 2, but the difference is, while in the case of anaphase 1, the force was derived from the kinetochore microtubule, while in the case of anaphase 2, the force is derived from the sliding movement of polar microtubule. And the there is another pulling force is being provided by the astral microtubule. So there is a no role of kinetochore microtubule as far as anaphase 2 is concerned, which is predominantly found in anaphase 1. Hence, the option A is correct, that the depolymerization of microtubules, that is the kinetochore microtubules, actually causes the separation of chromosomes, which allows these chromosomes to move towards the poles. Which of the following is observed during anaphase B? Depolymerization of microtubules, shortening of polar microtubules, consumption of ATP, or sliding of kinetochore microtubules. As we have just discussed in the previous slide, that during anaphase A, kinetochore microtubule depolymerization and gets shortened, which ultimately generates the phosphor chromatid separation. During anaphase B, polar microtubule grows, not shorten, and slide past each other, which actually causes the chromosomal segregation. Unlike anaphase A, anaphase B is more dependent on nucleotide requirement and especially heavily dependent on ATP. So, in this case, the answer C should be the correct answer, that anaphase B, which is heavily dependent on ATP consumption. So, option C is the correct answer. If the sequence of the coding strand in a transcription unit is as follows, where the sequence is given, the sequence of mRNA transcribed from the transcription unit would be. So here there are 4 sequences which are given, and we have to predict what would be the sequence of mRNA if the sequence of a coding strand is given. See, always remember, as a general rule, whatever is the sequence of your coding strand, as a rule, that would be the sequence of your mRNA, except that thymine would be replaced by the uracil. So here the option A would be correct, that the coding strand sequence, if you can carefully see, that is the sequence which is present in option A, except thymine is replaced by the uracil. So option A would be the correct answer. If the sequence of the coding strand in a transcription unit is as follows, the sequence is given. Again, with the same analogy as we have just previously discussed, the coding strand is being given and we are being asked what is the sequence of mRNA. As we have just discussed, जो भी आपका कोडिंग स्ट्रैंड का सीक्वेंस है वही एग्जैक्टली एमआरएनए का सीक्वेंस होगा, except thymine is replaced by the uracil, and by same analogy the option C should be the correct answer. The variant hemoglobin in sickle cell anemia binds oxygen less efficiently. However, the abnormal allele has not been eliminated from the affected population, as it has a selection advantage against. That's very easy. We know that the people who are suffering from sickle cell anemia, they can better adapt in those areas which are prone for malaria. So option C is the correct answer. Ampicillin inhibits bacterial DNA synthesis, RNA synthesis, protein synthesis, or cell wall synthesis. Basically, ampicillin inhibits bacterial cell wall synthesis. It does just by inhibiting the transpeptidase. Basically, ampicillin is a competitive inhibitor of transpeptidase, which is required to make the bacterial cell wall. So hence, the option D would be the correct answer. Marsupial mammals moved from South America to Australia via Antarctica, Madagascar, the Galapagos Island, or Africa. See, DNA evidence, they suggested that the marsupials must have originated in South America, and in history they must have migrated from South America to Australia through Antarctica. So option A is the correct answer. The species inhabiting in different geographical regions are known as allopatric species, sibling species, sympatric species, or biospecies. Allopatric species are the species which have been isolated from other group of members due to the geographical isolation. That means an extrinsic mechanism like a geographical isolation is playing a dominant role to divide a group of people into 2 different species. Now one group of people cannot mate with other group of people because of the difference in the distance, and hence allopatric species is the correct answer for this question. In ecological succession from pioneer to climax community, the biomass will decrease continuously, increase and then decrease, decrease and then increase, or increase continuously. Whenever community move from pioneer to climax community, definitely the number of individuals are going to increase, and hence the biomass will also increase. So in this case, the option D would be the correct answer, that whenever any pioneer community becomes a climax community, the biomass definitely increase continuously. The alarming rate of depletion of biodiversity in recent years is mostly due to the global warming, habitat destruction, pollution by pesticides and heavy metals, or ozone depletion. As we all know that habitat destruction is the primary cause for the loss of biodiversity, while exotic species invasion is the secondary cause of the biodiversity depletion. Interestingly, if you see, that once on this earth, 14% of the earth's surface was covered by the tropical rainforest. And because of the heavy habitat destruction, now we are left with only 6% left of such tropical rainforest. So hence, in this case, habitat destruction is causing the alarming rate of depletion of biodiversity. Nucleosome core is a structural unit of chromatin consisting of, that's very simple. Nucleosome, it is the basic structural subunit of a chromatin, which consists of 200 base pair of DNA around and an octamer of a stone protein. So option C would be the correct answer in this case. Repeat core sequences consisting of 2, 3 or 4 base pairs are known as single nucleotide polymorphism, microsatellites, mini satellites or telomers. Single sequence repeats or microsatellites, they are the DNA stretches which consists of short tandemly repeats of di, tri, tetra or even penta nucleotide motifs. So hence the option B would be the correct answer. Which of the following genotypes causes Klinefelter syndrome? XO, XSY, XX and XYY. The genotype XXY, it causes the Klinefelter syndrome, while XO, it causes the Turner syndrome. So hence the option B would be the correct answer. When a heterozygous dog mated with a homozygous recessive parent, what will you describe this as? Test cross, reciprocal cross, monohybrid cross or a dihybrid cross? If you can see in the figure, there is a cross made between a heterozygous dominant condition as well as homozygous dominant condition with their respective recessive parent. This type of cross is known as test cross, and it is being done to know the identity of the heterozygous or homozygous dominant parents. If the population, the F1 progeny, is having the 50% of the dominant as well as 50% of the recessive character, then one of the dominant parents would be the heterozygous conditions. While if the F1 progeny is having all dominant character, then the parent which is having a dominant character must be a homozygous condition. So hence, whenever a heterozygous condition is mated with a homozygous recessive condition, then such cross is known as test cross. A gene showing codominance has one allele dominant to other, both alleles independently expressed in heterozygotes, allele tightly linked on the same chromosome, alleles expressed at a same time in development. This is a very simple question. Phenomena of expression of both the alleles in heterozygous condition is called as codominance. For example, in a blood group system, IA and IB, whenever they are present in both together, then the blood group is known as AB rather than A or B type. So hence the option B would be the correct answer in this case. The transition from water to land in the evolution of vertebrates occurred during Cambrian, Jurassic, Carboniferous or Devonian period. As you can see in the diagram, that water to land transition, it must have occurred almost 400 millions around ago, and which is a time period for the Devonian period. Hence the option D would be the correct answer in this case. Okay, in this question there are two questions which have been asked. Maximum diversity can be seen in, that's very easy, tropical rainforest, they have the maximum diversity. Since there is a plenty of diversity is there, that simply means there is a plenty of food is there, and hence there will be more animals which can be sustained in such forest. So in both the question, the answer has to be option A, that is tropical rainforest. Passive immunity is obtained by, first of all, we should know what is passive immunity. See, there are two types of immunity is there, active immunity and passive immunity. Active immunity क्या होती है? ऐसी immunity जो हमारी बॉडी खुद डेवलप करे, either through variolation और vaccination. While पैसिव इम्यूनिटी क्या होती है? जिसमें हम प्री फॉर्म्ड एंटीबॉडीज अपनी बॉडी के अंदर लेते हैं, मे बी थ्रू मदर और मे बी थ्रू एंटी सीरा, लाइक एज वी टेक एंटी टेटनस इंजेक्शन और एंटी वेनम इंजेक्शन. सो पैसिव इम्यूनिटी के एग्जांपल क्या होगा? पैसिव इम्यूनिटी इज ऑब्टेन्ड बाय इंजेक्टिंग द सीरम ऑफ अनदर एनिमल ऑब्लिक इंडिविजुअल कंटेनिंग एंटीटॉक्सिन. सो ऑप्शन ए इज द करेक्ट आंसर. अब इन दोनों ही क्वेश्चंस में आप देखिए, दोनों ही क्वेश्चंस में पैसिव इम्यूनिटी पूछी गई है. बेसिकली इम्यूनिटी एक्वायर्ड बाय इन्फेंट फ्रॉम द मदर थ्रू मिल्क, दैट इज एग्जांपल ऑफ पैसिव इम्यूनिटी. मदर के मिल्क में ऑलरेडी प्रीफॉर्म्ड एंटीबॉडी है जो कि फीटस को या फिर चाइल्ड को दी जा रही है. while injection of antivenom, abhi we have discussed that it is artificially acquired passive immunity. So in both the cases the answer has to be option B. The predominant antibody isotype released during primary immune response is, that's very easy. हमेशा याद रखिए, प्राइमरी इम्यून रेस्पॉन्सेस में कौन सी एंटीबॉडी हमारी काम आती है? आईजीएम. जबकि सेकेंडरी इम्यून रेस्पॉन्स में कौन सी एंटीबॉडी काम आती है? आईजीजी. सो इन बोथ द केसेस द ऑप्शन बी इज द करेक्ट आंसर, दैट प्राइमरी इम्यून रेस्पॉन्स में आईजीएम और सेकेंडरी इम्यून रेस्पॉन्स में आईजीजी. तो ऑप्शन बी इज द करेक्ट आंसर. Which of the following immune cells are the most effective in destroying intracellular pathogen? See, for destroying intracellular pathogen, T cell, which is the T cytotoxic cells, are the most effective, while they are the least effective against extracellular bacterial pathogen. So, in this case, the option A, that is cytotoxic T cell, is the correct answer. Phagocytes do not swell in hypotonic solution. The most plausible explanation for this is absence of sodium channels, sodium potassium ATPase, aquaporins or potassium channels. होता क्या है? Phagocytes, they are generally laid in the water, and since they are do not having aquaporins through which water may take entry, इसीलिए generally वो swell नहीं करते. तो इस क्वेश्चन का आंसर होगा C, absence of aquaporins. Thank you for watching. Please do subscribe us and hit the bell icon for the more updates. Thank you.